All right, well, welcome everybody. Again, my name is Christy Sullivan, and I'm the Secretary of the American Society for Cellular and Computational Toxicology. This is the September webinar in our monthly series with ASCCT and the European Society for Toxicology in Vitro. And I want to encourage you to visit our websites to learn more about our organizations. You can join a mailing list to find out about events, and I would love it if you would consider becoming a member to support programs like this webinar series and our meetings and support for early career scientists and students. Just a couple of housekeeping items. This webinar is recorded, and the recording will be posted on the ASCCT website a few days from now. In the webinar platform, you can ask questions of the presenters using the Q&A feature at the bottom of your screen. There's a toolbar there. Today, we're going to have both speakers give their presentations and then have a joint Q&A afterwards. But you should feel free to just add your questions when you think of them for the speakers, and we'll save them. You can also vote or give sort of a thumbs up to some of the questions that you see there in case you really want to hear the answer to that question as well. And if you wouldn't mind, indicate which speaker your question is directed to. We have also enabled closed caption subtitles. If you want to view or hide them, you can do so at the bottom of your screen where you see live transcript. And finally, please say hello in the chat and let us know where you're joining from. So I just wanted to briefly announce or remind you of a couple of events coming up. First of all is the ESTIV Congress. That is going to be in November in Spain, in Barcelona. And registration is still open, so you can attend. You can actually also submit a poster abstract. Today is the deadline, so hop on over there if you want to do that. We also have the ASCCT meeting coming up. This is going to be next month, and actually one of our speakers is going to be speaking there, so we hope you will join us. There is in-person registration possible until October 7th. There is also online registration. Some of the sessions will be broadcast online, so you can do that if you aren't able to travel to North Carolina. Because of this annual meeting, we won't have a webinar next month. So hopefully you will attend the meeting and view some of the great talks that are going to be happening. You can just scan that QR code on your screen to get to the meeting website and find out all the information you need. And also the NACEDM is hosting a workshop and some webinars leading up to this. This is on using new approach methodologies to address variability and susceptibility across populations. So the workshop is the 26th and 27th, and I will pop that link. I know that's a long web address. I'll pop that in the chat when I'm done speaking. But in preparation for that workshop, there have been a series of webinars which you can view on the PCRM Neura page. And there is also one coming up October 7th, talking about chemical exposure in susceptible populations. So finally, on to our speakers for today. Both of our speakers are going to be talking about important concepts on the road to transitioning to greater application and use of new approach methodologies. Our first speaker is going to be Dr. Katie Paul Friedman. She joined the Center for Computational Toxicology and Exposure in the Office of Research and Development at the US EPA in 2016. And she focused on the application of new approach methodologies to assess chemical safety with additional interest in variability in endocrine bioactivity and alternative and traditional toxicity variability and developmental neurotoxicity. One of her roles in the center is to run the Kass program. And previously, Dr. Paul Friedman worked as a regulatory toxicologist at Bayer Crop Science and received her PhD in toxicology from the University of North Carolina, Chapel Hill. And our second speaker is Dr. George Kass. He was trained as a biochemist and received his PhD in biochemical toxicology from the Karolinska Institute in Stockholm. After postdoc at the Swiss Federal Institute of Technology in Zurich, he returned to the Karolinska Institute, then moved to the University of Surrey in the UK, and then finally to his current home, the European Food Safety Authority in 2009, where he is lead expert in toxicology. He's published over 140 papers in the field of toxicology and chemical risk assessment, and his work focuses on the molecular mechanisms of drug toxicity and liver injury. In 2020, he was elected to the Académie d'Agriculture de France. Okay, so I'm going to stop sharing my screen and give the floor over to our first presenter, Dr. Paul Friedman.
All right, thank you, Christy, for the introduction. And thank you everyone for joining. We have a really nice crowd. All right, can everyone see my screen okay?
Yep, looks good.
All right, as Dr. Sullivan stated, I'm Katie Paul Friedman. I'm a toxicologist at the EPA in the Office of Research and Development. And today I'm going to give you a talk about performance expectations for new approach methods, or NAMs, in predicting the effects from repeat dose animal studies. And I have to give you a little bit of warning that these are my views and not the views of the US EPA. So to get started, why would I be talking about traditional animal data and the variability in traditional animal data when really what we're talking about in this session is acceptance of new approach methods? And I'm going to give you a little background on why this is important. Essentially, the variability and the performance of in vivo traditional methods and the comparison then to in vitro methods is one component of evaluating the performance of NAMs. It's part of validation, it's part of scientific confidence. And so that's why we're examining this comparison between how variable in vivo methods are and how well in vitro or in silico methods might perform in comparison. And to do this, I'm going to highlight some published work from our group and then some ongoing work in 3 parts. So in part 1, I'm going to talk a little bit about a paper from 2020, Pham et al., where we looked at benchmarks on quantitative reproducibility of systemic findings in repeat dose animal studies. So these are subchronic, chronic, multi-dose studies, typically in rats, dogs, mice, rabbits. Part 2 is work that was led by Prachi Pradeep, who was formerly a postdoc in our group, and she is now actually on this call. Thanks for joining, Prachi. And in this work, what we did was incorporate estimates of variability in those repeat dose animal studies into a quantitative structure activity relationship model for prediction of repeat dose point of departure estimates. And so you can see the relationship between defining the variability in vivo studies and then using that expectation of variability to consider a point of departure estimate from a QSAR as a distribution. And maybe that's a little bit more realistic than a single point estimate. And then in part 3, I'll give you a little bit of a preview of some work we have in progress right now, hoping to finish up on the end of this year, where we're looking at benchmarks on the reproducibility of organ level findings. It's a little bit more of a biological phenotype and how reproducible that might be across species and across studies for a given chemical. And then some conclusions on how this variability information informs our expectations of NAMs moving forward. So to step back a little bit into the background, in the US, we have the Toxic Substances Control Act, and there's this text in there. It's very similar to some text that's in REACH. It's similar to some texts in other regulatory frameworks. It essentially says that we can use NAMs to the extent that they're practicable and scientifically justified, and that they perform at an equivalent or better scientific quality level than the traditional animal methods. And so what you're left with when you read this is, well, how well did the traditional animal methods work in terms of predicting a point of departure? How reproducible were they? How variable were they? And so that's why we initiated this work. And so from our perspective, essentially, we can define these expectations of NAMs, whether they be in silico, in chemico, or in vitro, for predicting repeat dose toxicity by examining how well replicate animal studies actually reproduce themselves in terms of a quantitative point of departure, as well as observation of a particular phenotype or target organ effect. And when we think about expressing variability, we read about it in the literature, sort of using in vivo data as reference data or training data to evaluate NAMs. We often see what I'm showing on the right of this slide. We often see truth kind of reduced to a qualitative evaluation of whether or not a chemical in a particular test was positive or negative. So you can see we kind of have the truth in quotes as traditional toxicology. We have a negative or a positive, and then we have some NAM-based prediction, and we reduce that to a negative or a positive. And then we look at this confusion matrix in order to calculate balanced accuracy, specificity, and sensitivity. We can also think about what I'm showing on the left of the slide in terms of quantitative variability. And this is really the spread of values. So if I had multiple, say, subchronic studies of a single chemical, would the point of departure from those replicate studies fall into a distribution like the blue one, where the values are very close together? Or would they fall a little bit more spread out, more like the green distribution? And in our part one, this is essentially the question we're tackling is a distribution of point of departure values for chemical based on replicate repeat dose studies. Is it a broad distribution or a narrow distribution? And to do that, we use a publicly available data source that we've developed within the center. It's called the Toxicity Reference Database, or ToxRefDB. This is available. You can completely download the database at this DOI. And what it contains are a number of curated studies from repeat dose animal studies. So chronic, subchronic, developmental, subacute, multi-gen reproductive, and then a small number of other types of studies. Typically, these studies are either guideline adherent or guideline-like for the 870 series health effect guidelines at the US EPA. And the study source for most of this curated data are Office of Pesticide Programs, OPP, data evaluation records, these DERs. And these are a really amazing resource put together by this office at the EPA, where they review data submitted by a registrant on a particular pesticide active ingredient. And they write narrative summaries of those studies and include data tables. And it's such an amazing resource that we curate the data from those. And so most of the database comes from there, but we've also curated information from studies at the National Toxicology Program, from failed pharmaceutical compounds, some from the open literature, and then some other. And if you are interested in this resource, you can read all about it in our previously published paper that describes not only the data in it, but how it was constructed. So in part one, that was our data source. And what we sought to do was establish a benchmark on the quantitative reproducibility of systemic findings in repeat dose animal studies. And so today, in the interest of time, I'll focus on two primary research questions that we tackled. There's more in the paper. I encourage you to take a look at it. But primarily, we asked, what is the range of possible effect values in replicate studies for a given chemical? And then we use some statistical approaches to characterize that. The first is we use the residual root mean square error, or RMSE, as an estimate of the variance that's in the same units as the systemic effect level values, so log 10 milligram per kilogram per day. And this RMSE can be used to define what we call a minimum prediction interval, just essentially estimate range for some given model that might seek to predict that point of departure. The second question that we ask is, what is the maximal accuracy of a new model that attempts to predict effect level values for a given chemical? And to do this, we use the mean square error, or MSE, to approximate unexplained variance in replicate study values. So this would be variance that we can't explain with the study descriptors or study metadata, essentially. And I'll go a little bit more into that in the next slide. This unexplained variance essentially limits how much variability can be explained. So it's limiting the R squared on some new model that would use these same data as the reference or training set. So as promised, you can see here, this figure from the paper is essentially a conceptual explanation of how we've defined variance. So we look at replicate studies of a given chemical, and we say we observe some variance in either the lowest effect level, LEL, or lowest observable adverse effect level, low AL, and that's the total. And we can explain part of that variance using study metadata or study descriptors. So these are things like the chemical identity, the study type, the study source, the strain, the sex, the administration method, and then continuous variables, including the number of doses, the dose spacing, study year, and the percent purity of the substance tested. So these are things we know about the study that are standardized that we can use as input features to explain some percent of the total variance. And then what's left over is approximated by the mean square error, and that's the unexplained variance. These are things that we didn't know to document, or they're just unknowns. And in this case, what we find is it's possible maybe there's some irreducible amount of error in the way that we run these types of studies across laboratories that we may not be able to get below a certain amount of unexplained variability. Now, mathematically, to work through this conceptual framework, we use two main approaches. We use multilinear regression, abbreviated as MLR throughout the slides. And here we aggregate all of the replicate studies at the level of a chemical. So the primary distinguishing feature of this approach is that the replicate definition is not stringent. We essentially take any study that was run, a repeat dose study in adults with oral dosing, and use it as a replicate study of that chemical that would be useful in defining a point of departure. This is really great in terms of the total N because it's maximized. So we maybe decrease the impact of outliers or database error rate, which of course will never be zero. But we make this assumption. We make the assumption that the study descriptors contribute independently to the variance. And to the extent that that's not true, we might overestimate the variance. And so we employed another approach as well to get a sort of range of understanding of how to approximate this variability. We call this an augmented cell means approach. And the primary distinguishing feature here is that the replicate definition is very stringent because we require the chemical, study type, species, sex, and admin method combination, that whole vector, had to be identical for two studies to be considered replicates. And because of this, our N is greatly reduced. And so that might bias the variance estimate because now our N is reduced by 90%. We're looking at a much smaller chemical and study space. However, this approach is better accounting for the possible interactions among descriptors. So for instance, we know that species and study type are not independent, right? If you have a chronic study in the US EPA framework, it's going to be in a rat, a mouse, or a dog. It's not going to be in a rabbit, right? So the study type and the species are truly linked. As an example. So here's our workflow. We took ToxRefDB version 2.0, where there's over 1,000 chemicals, and we've curated almost 6,000 study reports. And then we had two workflows. Part A is for the multilinear regression workflow, and part B is for that augmented cell means, more stringent definition workflow. You can see we had requirements on the studies saying that we're only looking at adults, systemic endpoints, oral, and milligram per kilogram per day dosing, or able to convert to that unit. And we end up with a full study set here of about 563 chemicals and a little over 2,700 studies. And then we actually do more modeling on some subsets of that data where we subset by study type. And you can see those data sets are even a little bit smaller, although for this kind of work, they're not tiny, right? Hundreds of chemicals and hundreds of studies. For the augmented cell means approach, you'll see the data set is so much smaller. So here we only have 96 chemicals and 278 studies. And if we subset that into study type, we start to get into some data sets where we're not sure if they're so reliable because it's just a really small. And so we have to have that limitation on our interpretation. So here, there's a lot on this slide. And what I want to do is boil this down to, we created 28 different models to approximate study variance using the approach I just described to you, because we didn't know which way was necessarily right. As I pointed out, each variance model had its own limitations and problems. And so we really wanted to understand what is a range, a reasonable range to estimate total variance in study level point of departure, unexplained variance, and then the explained variance or RMSE. And what you can see is the total variance, regardless of the model type we use, and regardless of whether we use the low ALs and low LALs for the full data set or by study type, the total variance tends to approach 0.75 to 1 log 10 mg per day squared. That's a really hard unit to wrap our minds around. We don't really know what that means as toxicologists to have a squared unit like that. It becomes more meaningful when we compare it to our estimates of the MSE, that unexplained variance. So you can see here, regardless of the model type we used and the data set we used, the MSE tended to be 0.2 to 0.4. So it's a fraction of that total variance. And on the next slide, I'll highlight this a little bit more, but essentially we see a third of our total variance that we can't explain based on curated features of the study. Now the RMSE, these are units we understand. So the actual unit of variance here is 0.45 to 0.6, so approximately half log 10 mg per kg per day. And we can use that to define a 95% minimum prediction interval. It's very similar to essentially using a standard deviation or a measure of a spread of the residuals to give you an approximate interval that a new prediction should fall into. So here you can see the percent explained variance. So this is essentially how much of that total variance could be explained by the study descriptors. You can see regardless of the modeling approach or the data set that we used, we tend to approach 55 to 73% that we can explain, which means that, there's maybe a third of the variance that we can't explain. This means that for some new model, an R squared on that predictive model might approach 0.55 to 0.73 as an upper bound on accuracy if it was trained using this data set. To translate this even further for toxicology, if you think about that 95% minimum prediction interval based on the RMSE, where would a reasonable new prediction fall based on the variability in replicate study data? Essentially, we're looking at plus or minus 1 log 10 mg per kg per day. So the example I give here is if you had some value that you believe to be the truth, it was 10 milligrams per kilogram per day is the point of departure. It's likely that given the variability in the reference data of this kind, that a model prediction of somewhere between 1 and 100 milligrams per kilogram per day would be the greatest amount of accuracy that you could achieve globally across a chemical set like this. And you can see, regardless of what model and data set you look at, essentially, it's a pretty wide interval, especially when you consider that for a lot of these studies, they might have only tested doses across maybe 3 orders of magnitude, from 1 to 1,000 mg per kg per day. So with that in mind, this next work that we published in 2020 sought to understand how to best construct an in silico NAM, a QSAR, for prediction of that point of departure. But now we have a little bit more of an informed expectation of what a good QSAR in this realm might look like. We might only explain 55% of the variability in the reference data. We might have a prediction interval that spans, plus or minus 1 log 10 mg per kg per day. So you start to think that what would be acceptable in terms of an in silico NAM is something that looks more like that. And that's what we evaluate in this paper. And Dr. Pradeep, evaluated several research questions. And here I'm going to focus predominantly on one of them, which is, can a QSAR from a QSAR point of departure value be represented as a confidence interval based on estimates of variance in the training data? And so what we did here was construct QSAR models using a point of departure distribution informed by the estimate of variance, and then bootstrap resampled that point of departure distribution to then represent a confidence interval around a POD QSAR value. So in brief, from the paper, essentially, we're trying to use this variability estimate to inform model uncertainty. You have a model that gives some result, in this case, a point of departure value, but it's an estimate of a true point of departure value. That true POD value, of course, is unknown. There's some variability and uncertainty in the reference or training data that will then propagate to variability and uncertainty in the model, and our estimate of how well it performs, right? The RMSE on the model, for instance, or the R squared. So what was done was a point of departure distribution was constructed for each chemical, where the mean is equal to the median experimental point of departure from the replicate studies, and the standard deviation is equal to 0.5 log 10 mg per kg per day units, based on our paper Pham et al. From 2020. Then 100 bootstrap resamples were built using random sampling of those point of departure values for each chemical to then form a pre-generated point of departure distribution. Then the predicted point of departure from this QSAR is the mean of those 100 bootstrap predictions, and the confidence interval around that estimate is plus or minus 2 times the standard deviation of the 100 bootstrap predictions. So now, instead of providing one point of departure estimate, there's a range, and we can compare that to the actual range that was observed across replicate studies for chemicals in the training set. And that looks like this from the paper. On the left is the training result, and on the right is the test result. And I want to point out right away some of the statistics on performance. So if you look at the RMSE and the R squared, now it starts to fit in, right? We've gone to great lengths to characterize the variability in the reference data. And it turns out that the RMSE on the training and test performance for the model really is not that much greater. The model is not contributing that much more variance than perhaps the data itself that was used in training. And the R squared, we're able to explain essentially a little bit more than 50% of the variability, which is within the target of just looking at the variance in the data itself from our Pham et al. Study. So you start to think, actually, this may be an upper limit on how well we can do in terms of repeat dose point of departure prediction using a QSAR. You can see here in the graph, this dark black line, diagonal line is unity. The red line is the predicted error. The horizontal line in black is the observed error from the chemical in training. And you can see now a little bit more, it feels a little bit more realistic, right? Because in some chemicals, you see a really wide observed error in terms of how reproducible the point of departure was across replicate studies. And so now it's a little bit more reasonable expectation on NAMs. And Prachi did a lot of work in this paper, and one of the other primary conclusions is from the enrichment analysis to evaluate the accuracy of the point of departure estimates coming off this QSAR. Most of the highly potent chemicals end up in the highly potent part of the prediction set. So 80% of the 5% most potent chemicals from the training set were found in the top 20% of the most potent chemical predictions. So this model is doing really well in terms of binning potent chemicals from less potent chemicals And it's doing pretty much as well as we might expect based on the variability in the training data. Part 3, I want to jump to our work in progress on benchmarks for qualitative reproducibility of organ level findings. We talked a lot about quantitative variability, but do we even identify essentially organ level effect across multiple replicate studies in the same organ? So here we have this value that I call percent concordance. This is essentially for a given chemical tested in multiple studies. Do I always find a positive in a particular tissue, or do I always find a negative in a particular tissue out of, the total chemicals tested? So you could have a chemical that's all positive or a chemical that's all negative, or you could have a chemical with mixed results across its study set. So here in the graph in the lower left-hand corner, you can see percent concordance. And then the x-axis, we have different organs: adrenal, kidney, liver, spleen, stomach, thyroid. These are the most commonly reported organ level effects from our repeat dose study set. So that's why we chose to examine these organs. And then we're color-coded by sample size, and we're subset by species and study type: chronic, subchronic, or all is combined for all species. And then we have dog, mouse, rat. And what we learn is, depending on how you group the data, the qualitative reproducibility of the organ level effect findings is highly variable. So it's 33 to 88% concordant, depending on how you actually group the data. Importantly, the within species concordance tended to be greater than within, say, study concordance. So you could have subchronic studies in multiple different species. So here, this round circle, these are the subchronic. So you have multiple species here, and that concordance is not as great as if I just look at, say, these triangles, and that's dog. It has a smaller N, and it's one species, and the concordance is maybe higher than, say, if I group all of the data together for all species and all study types. Now my rate of qualitative concordance is probably lowest, right? Greater concordance was observed in organs that have higher negative rates. So for instance, stomach doesn't have findings reported quite as often. It tends to be negative, and it had some of the higher concordance values because a lot of the chemicals were negative. You can see here, it's very easy to get a sense that across species, things are less replicable, and that's really consistent with what's been reported in the literature previously. But it starts to inform, if you reduce qualitative reproducibility to a binary, negative or positive, we have a lot of variability in what could be negative or positive. How would you classify these chemicals as negative or positive for a particular organ? The other thing I wanted to pull out from our unpublished work is thinking about how many NAMs we will need to capture something like a qualitative effect at a target organ, whether liver is implicated or kidney is implicated. And so what we did here was ask the question, can target organ effects be identified using just a subchronic study? What are the odds that a chemical will produce any organ level effect in a chronic study if the subchronic was negative for that organ? So to do this, we used odds ratios for matched chemicals where we had a chronic and a subchronic study for the chemical, and tried to understand whether or not we would miss anything if all we had was the subchronic study. And what this doesn't speak to is adversity. We're not looking for necessarily a cancer finding that you wouldn't see in a subchronic study. We're looking for histopathological changes, weight changes, any kind of change at the organ level, regardless of adversity. Would we have identified an effect in the organ? So here, organ is color-coded, the same 6 organs that are most highly represented in our positive findings for repeat dose studies, and then species where we've either combined all or broken them out. And what you can see is the odds ratio of a positive at a given tissue in a chronic study, given a positive in the subchronic. All of these odds ratios are over 1. So if you find a histopathological change in organ at the subchronic, you're much more likely to find a change in that tissue in the chronic, anywhere from, just greater than 1 to, for some tissue and species combinations like this one, this is dog. You can see in the dog, you're, maybe 5 times more likely to see a positive in the chronic if you saw a positive in the subchronic for the adrenal gland. Conversely, and maybe more importantly for how we would consider these data in terms of alternatives and replacement with NAMs, is that the odds of a positive in the chronic study in a particular tissue given a negative in the subchronic are all below 1, which means it's not likely to observe that case. So the possible indication here is that a repeat dose point of departure for a target organ at 90 days, particularly for liver and kidney, where we have a lot of data to support this, it's likely protective for a chronic finding on a dose basis. So while we might not see the level of adversity with a shorter duration exposure, we would be able to identify some effect in that tissue and identify a point of departure for that tissue that would be likely to be protective. So in conclusion, how does this compare to other work in this area? There are some previous QSAR models of subchronic oral rat, NOAEL values, some other sort of chronic and subchronic studies. And this is nicely summarized in this table from Pradeep et al. 2020, where we looked at essentially other studies that have used similar data sets, repeat dose studies in rodents typically, and built a QSAR. And you can see the numbers of chemicals from hundreds to a few thousand, and then what RMSE they report and what R-squared they report. And you can see that, our estimates of variance and repeat dose data, even from different sources and of slightly different types, seems to be fairly on par with the limited research out there, that RMSE may approach a half log 10 mg per kg per day in a lot of data of this type. And that explaining 50 to 70% of the variance in those data is actually pretty good. So the primary takeaways from this whole sort of storyline that we've been working on in the center for a while is that we should have more informed expectations of NAMs to replace repeat dose studies. So the variability in vivo toxicity studies that we're using in either training or evaluation of NAMs will limit the predictive accuracy of those NAMs. And so in this case, the prediction of a systemic effect level within plus or minus 1 log 10 mg per kg per day fold of some true value, it's actually very good in silico NAM. And it's really important for the acceptance of NAMs for chemical safety evaluation that we understand that we're dealing with a highly variable value to be predicted, let alone the variability that would be introduced by the model itself. In part 2, we demonstrate how that might work for a QSAR. And then in part 3, we start to look at some qualitative reproducibility issues at the organ level, looking for any organ level effect observations and trying to understand how reproducible those might be depending on how you group data, and how we might be able to develop protective and predictive NAMs by grouping the subchronic and chronic data together, or by only having a subchronic study and not necessarily needing a chronic study. Finally, my main conclusion is that it seems likely we could construct NAM-based effect level estimates that might offer an equivalent level of public health protection as the effect levels that are produced using animals. And so if we're able to continue building scientific confidence in those approaches, we might be able to provide a bridge to a major reduction in the use of animals, and perhaps identification of cases where an animal study might provide scientific value. With that, I want to thank the center and especially thank Richard Judson, Woody Setzer, Lily Pham, Prachi Pradeep, MJ Foster, Sean Watford, and Rusty Thomas, who are all co-authors on a number of these works and have contributed so much scientific thought to this area. So thank you.
Great, thank you, Katie. That was great. I really like the whole storyline because you're putting a lot of these questions into context. and making sure we're keep being aware of what our current system provides. And then I like this, the end part I haven't seen yet, but I really like that because we're thinking about on the road to replacement and then shorter term reductions. So good stuff.
Thanks, Christy.
Well, let's go to George. Dr. Kass, are you ready to present?
Okay, I am. Thank you very much. Let me first try to share the screen. It should be the one here. No, sorry, wrong one. This one. Okay, can you see the screen?
Yeah, looks great.
Okay, great. Thank you very much, Christy. And yes, good morning, good afternoon, good evening from my side. First, I would like to say thank you to the organizers for inviting me to contribute to this webinar. I'll be sharing with you some of the work that we've been doing at level of EFSA to bring in the NAMs into the regulatory context. I'll be sharing with you the opportunities that are presented, but also some of the challenges and problems we face at the European level for using NAMs to replace or at least to complement in vivo studies. Let me start, it's not working yet. Here we go. Yes, so first of all, I need to also make a disclaimer. So views and thoughts and opinions are mine and not necessarily those of my organization. So EFSA is one of those EU agencies. We're not based in Brussels, where the Commission is, but we're spread in Europe. And what you can see here on this map is, first of all, our location. So we are in the north of Italy. Let me see if we make a pointer, laser pointer. So we are here in the north of Italy, south of Milan and north of Bologna. And it's one of the agencies that in Europe that deals with safety of chemicals, but also safety of microbial agents. We're dealing with the food aspect of it, whereas the colleagues in Amsterdam, in AMAD, deal with the pharmaceuticals, both for human health, but also for the veterinary drugs. ECHA, on the other hand, deals with industrial chemicals. And also in the north, we have ECDC, which is primarily focusing on diseases. So zoonoses and aspects related to human health are very much their portfolio. The areas that EFSA is tackling, and this is just to illustrate or to introduce where the chemical risk assessment comes in and the problems we have in using NAM approaches. So we're dealing with what we call from the farm to the fork, and this takes us really from the plant health, plant protection, GMO. We're dealing with animal feed. We're dealing with animal health and welfare. Also biological hazards. So zoonoses such listeria, for example, are issues that we deal with. We deal with chemicals, so contaminants, as well as chemicals that are intentionally put into food, such as food additives. And we also deal with chemicals that can come into the food because they're used in containers, in packaging materials. And finally, we also have nutrition as part of our remit because it addresses aspects of vitamins, oligominerals, main macronutrients such as carbohydrates in the context of human health. This is just to illustrate that for the majority of the sectors that EFSA is working, we're dealing actually with chemicals and chemical risk assessment. So as I said already, the types of chemicals we get can be so-called regulated, so intentionally put into food, and there normally we have applicants that provide the information to support the support the risk assessment. Whereas when we're dealing with contaminants, generally there's no commercial producer behind it. So to be able to, the data we can get for doing the risk assessment comes generally either from legacy data or from data from published papers. And finally, we're also dealing with chemicals that are already on the market, that are authorized on the market, but they have to undergo a reevaluation. This is typical for plant protection products in the EU. The type of data and the amount of data that are being submitted to EFSA depends very much on the legislation. And I've got two examples here to illustrate it. So we have the legislation on the top here, which is dealing with the plant protection products that are on the market, which is very prescriptive. And we see that in a moment in terms of what data are required. Whereas in the legislation dealing with food additives in the EU, the legislation simply says that the food additives have to be safe for human consumption. And the responsibility for doing the risk assessment is down to EFSA, that produces a guidance informing the applicants what type of information they have to submit for us to do a risk assessment. So we have quite a divergence of approaches. One where the tests are being specified in the regulation, and I'm going to show this for this example for the plant protection products. So what is the legislation says? The information will be sufficient to evaluate foreseeable risks. And you can see in the legislation in one of the appendices, section 5, these are the studies that are requested by legislation to be submitted to EFSA for the evaluation. And you'll see immediately that the vast majority of information that is requested comes from in vivo studies, so studies carried out in experimental animals, where there's only a minor proportion that comes from in vitro studies. So although we have the possibility in the other sectors to specify if we are in a position of or have the possibility of devising guidance documents, we have the possibility of specifying which types of data are submitted. Generally speaking, we're still at the level where the biological studies that inform us on the behavior of the chemical, so the ADME information, as well as the toxicological studies. So we're dealing with the subchronic, chronic, reproductive developmental studies. All are from animal studies, and they all are requested to use the prevailing OECD test guidelines and using GLP criteria. For in vitro and using NAMs, what we have, in fact, is limited to genotoxicity and metabolism data. Again, of course, they have to follow the OECD test guidelines and GLP criteria. So the take-home message here that the traditional chemical risk assessment, as performed in the EU, still mainly relies on animal bioassays. So, looking at the past two decades and see how EFSA have been looking at at alternative approach and making use of alternative approaches in the chemical risk assessment. First of all, we very much focused on genotoxicity testing, where our guidance document, in fact, has established a battery of in vitro tests, and when there's clear evidence of genotoxicity, there's no need anymore for in vivo tests. We've also been using the TTC approach, So the threshold for toxicological concern for chemical risk assessment, was readily used when EFSA in fact started, or shortly after it started. So we're talking here about 2004 with an approach that was used for flavorings. It's been used for the risk assessment of some impurities, metabolites, and certain degradation products. It's been used for pharmacologically active substances present in food of animal origin, and also in the combined exposure to multiple chemicals. And we have recently published a guidance document for how to use the TTC approach in chemical risk assessment for EFSA. We've also been using the read-across approach, again, to avoid the testing of the chemicals in animals. So it's been applied to flavorings, and also applied to the combined exposure to multiple chemicals. So we recently published a guidelines or guidance on the grouping of chemicals. And also for food contact materials is also an area where we use read across. It's more there on an ad hoc basis. We also been proposing non-animal tests for the establishment of residue definition for dietary risk assessment. So here it is the area here is the plant protection products and their residues. And one of the issues one has in plant protection products is that the number of metabolites that can be produced from an active substance can be extremely large. So we're not simply dealing with a metabolite that is produced or would be produced in humans or would have been produced in the animal studies, but we also have to look upstream of this, where the plant protection product or the active residue, the active substance, will already be metabolized by the plants. It will be metabolized by the microbes in the soil. There will be some degradation products for UV light, etc. So a single and a very simple chemical that is sprayed on the crop can end up as multiple metabolites. Of course, not all metabolites will be present at a very high level. Some will be present in very low levels, and this is the reflection on the approach for residue definition. And in this slide would be it's just to illustrate what we do for these metabolites of these plant protection products. And I should also mention, which is very important, some of those metabolites cannot even be synthesized. So there's really also a technological problem in actually testing them. So we used here an approach, and this is for the genotoxicity assessment, of combining read-across and the TTC approach. So these are an illustration of part of our activities already over the past decade, really bringing in non-testing approaches into chemical risk assessment. But now I want to spend some time on really what's happening now in the year 2022 and where do we see ourselves going in the future. Well, one of the important developments that has been happening in the EU level, at the EU policy level over the past 2-3 years is the Green Deal. And subsequent to the Green Deal, there was a publication of the Chemical Strategy for Sustainability towards a Toxic-Free Environment. And the CSS document, as we call it, specifies very clearly that the chemical risk assessment and safety testing needs to become more innovative in order to reduce the dependency on animal testing, but also to improve the quality, efficiency, and speed of chemical hazard and risk assessments. And it's clearly mentioned that the Commission will also foster research to move away from animal testing. This is also captured at the level of EFSA, so EFSA's policy in this sense. So it is the strategy which was published last year, so the 2027 strategy. And this strategy also very clearly specifies that EFSA will develop and integrate new scientific developments focusing on NAM-based methods and the minimization of animal testing. And one of the key actions is really to develop and integrate new approach methodologies and omics for regulatory risk assessment. So we're really very much moving in the same directions as in the US, really bringing on board new approach methodologies in the risk assessment. So how do we see this? First of all, we are currently developing a guidance on the reuse of read-across protein food safety assessment. We did not have a guidance so far. And what is important there? First of all, we were looking at the regulatory applicability of the read-across to chemicals, the testing opportunities for biological read-across, and also how we could use NAMs to underpin the read-across. We're currently working in parallel, the guidance development is ongoing in parallel with a big project that is also, like what Katie has been showing, is also focusing on plant protection products because of the high data density and also the high data quality that comes in this sector. So in fact, we're now having a big project taking our database on plant protection products, looking at all the endpoints, looking at all the metabolites in the context of repeated dose studies, and see how we can test the read-across framework or the guidance that is currently under development for this type of for this database, to see how applicable it is for our purposes. We also taken the opportunity, as we've been developing over the past few years, new guidance documents, to really look very closely at where NAMs can be applied. Often and for most of them, of course, we still have a requirement for in vivo studies, but really what we want to emphasize is the opportunities for using NAMs. For example, this one is for this guidance for nanomaterials, and we emphasize that the in vitro test, NAMs tests, will play an important role for characterizing or getting better insight into the hazard of the nanomaterials and their mode of action. Also the internal exposure, and looking very much more closely at what is happening at a cellular level and the fate of those nanoparticles, but really can only use in vitro approaches to get answers to those questions. We also develop, so in that guidance, advocating the use of in vitro studies to get more mechanistic information on the toxicokinetics and toxicodynamics of the nanomaterial, of the nanomaterials. Another area where we've invested considerably is in the area of developmental neurotoxicity. A lot of that work has been done in very close collaboration with the OECD and some universities in the EU. And this was driven not only by the fact that the animal models for DNT are not very good, but also this was seen as an opportunity to develop an IATA that can be used for testing the safety of chemicals. And as you can see on the left-hand side, there's a testing battery which has been developed, and we tested it on a number of case studies. And this IATA for us is very promising for the future implementation of NAMs. Another area that often comes up in chemical risk assessment when dealing with animal data is that of non-monotonic dose responses. The challenge that is often faced is that animal studies use a reduced or a small number of doses, and occasionally the dose response, the points for the dose response tend to provide rather unusual shapes, and not your classic sigmoidal shape, often due to the fact that we're dealing with just too few doses to come to conclusion. So it's difficult really to say that a chemical is having a different behavior and could be non-monotonic, or whether there could be a low dose effect simply based on the traditional animal testing with only a few doses. And for this, in our guidance document, so in our opinion, it is really we advising that that this can be only resolved using NAM approaches, where we can get more mechanistic understanding of really what is happening, what is really happening, and it's much more much easier to devise a testing strategy, a NAM testing strategy, where you can test a wider spectrum of doses. Also, an area we also advocate the use of NAMs is in the grouping of chemicals. So the approach EFSA has taken for the grouping of chemicals for the risk assessment of combined exposure to multiple chemicals is very much driven by MOA and AOP, so mechanistic considerations. And of course, for this, to get this integration of the data and the grouping, we really need NAMs to help us to bring this together, so that we can really classify chemicals and group chemicals based on solid MOA or AOP evidence. We also, as an organization, because we do not have any laboratories, so we do not have any facilities to conduct any experimental work, we tend to outsource most of our work, all of our work in that sense. And I think it's fair to say that the biggest chunk of the work that EFSA has been outsourcing, so the support that EFSA has been given for projects outside its organization, has been focusing on NAMs, and particularly on case studies, really to gain the confidence in using NAM approaches for risk assessment, and building up that necessary confidence. Pesticides, we have a new project on it's ongoing on neurodegenerative diseases. So as an example, Parkinson's disease. We're focusing on nanomaterials, but we're looking at GI uptake and genotoxicity using NAM approaches. We have projects looking at what artificial intelligence and machine learning can contribute to NAMs, how they can support it. We have a project that's just started on the immunotoxicity of the polyfluorinated compounds, or the PFAS. We also have a project on essential oils. So this is not for human safety, but is it for animal safety? So in the feed sector, feed additives, and we use NAM approaches to look at interspecies metabolic differences. We also build up, and this is thanks to the work by Jean Dorn, who's been very instrumental of putting it all together. So our TK plate is now 2.0, which is an open source platform where a lot of the efforts also been focusing on the PBTK models. And of course, for us, it would become very important to really understand and to model the behavior and the fate of chemicals, so that we can have a way of doing the reverse extrapolation from in vitro data, where we can get a point of departure in vitro, but extrapolate, working out using the proper models, QIVV models, to be able to predict what this corresponds in terms of exposure, external exposure, because from a regulatory point of view, it's important to be able to regulate external exposure. And another project which has just started is framing the human variability in toxicodynamics. So traditionally, we've been using a factor of 3 to do the interhuman variability for the toxicodynamic side. But here we have a project looking at this over larger populations, and based on QAOPs. We also have, as part of our roadmap on NAMs, we have a number of projects that are currently out there for tender. So these are calls for new projects, AOPs for developmental neurotoxicity, framework for metabolic syndrome, AOP for stress response, and also glucocorticoid production, NAM transcriptomics to predict target organ toxicity. So these calls are still open. So if you're in Europe, please take a look at this, although the deadline will be approaching pretty soon. We also have now a project where the tender is now closed, one on ADME to define which in vitro models can be used for predicting metabolism, human metabolism of compounds, and also another project for how to use NAMs for or how to integrate NAM results for chemical resistance using nano compounds, nanomaterials as case studies. This is sort of our current view and what we're hoping would be happening over the future, the coming few years in terms of the risk assessment. So what we really want to see is the classical, in terms of the hazard identification and characterization, to have the in vivo tox box shrinking, being slowly replaced by what we would call the NAM data galaxy. What we also want to see is beyond the use of NAMs, it's really trying to get a bit more, much more holistic approach to the risk assessment, to the risk characterization. Well, we will try to have a much more, a more holistic view for the exposure side, so not just based on consumption or occurrence data, but also taking advantage of the exposome data to work out what are the compare the dietary exposure with non-dietary exposure, internal exposure, but also bring on board biomonitoring data to better understand the exposure, and then with the help of TK data, really trying to link the hazard side with the exposure side, and also making more use of epidemiological data. And then, of course, none of that work could be done or should be done alone. So we should not be working just with ourselves here on those projects, but we have a very large network of long-established partners in terms of collaborations, or some are more recent, as you can see. So we work closely with URL ACFAM, the JRC lab in ISPRA. We work very closely with ECHA, who are developing similar approaches for implementing NAMs in the risk assessment. And we've been working very closely with a number of European funded projects. EU Tox Risk, which just finished now, but we have the new ASPIS consortium with Risk Hunter, OnTox, Precision Tox. We're collaborating closely in those projects, and we also partners in the new partnership that has been launched under the European, under Horizon Europe, so the partnership for the assessment of risks from chemicals, so-called PARC. But of course, not just in Europe, we work very globally. We're involved in APCRA. We work closely with OECD, WHO, FAO, the Global Coalition for Regulatory Science Research, food agencies across the world. We also have our own network in Iraq for sharing information on risk assessment methodologies. And just want to finish with a couple of thoughts, and really how we see NAMs and still some also the opportunities and challenges and the vision we have. Challenges, of course, are very much illustrated here. Really, we need to deal with high quality NAM data using benchmarked and robust tools. We need to have the confidence in using them. We need to know what their coverage is in terms of adversity. And also we need to learn how to interpret the data. Hazard data from NAM data are different, or maybe different from hazard data you get from in vivo studies. The information needs to be integrated in usable form. And of course, we need to build up the next generation of risk assessors through training. And of course, as I said already, you cannot do this on your own, and it shows it's not a triangle between regulators, academia, and industry, but there are four pillars here, really, because we also need to consider society. Society also has to accept what we're trying to do to ensure that they feel confident that they're still being that safety of society is maintained. And of course, it needs a lot of funding. We're doing our best to support projects, but also a lot of ongoing projects in the EU that I've just mentioned that have as a goal really to push forward the NAM sector, really to bring them into regulatory context, bring them into the decision-making process for chemical risk assessment. And with here I stop. And thank you for your attention.
Great, thank you, Dr. Kass. That was a really nice overview, and I especially like that diagram where you showed kind of epidemiological data coming in, and I think it's nice to see sort of a more expanded view, of taking a look at all of these types of data. So we have lots of questions coming in. We have a nice chunk of time for some Q&A. So let me encourage you to, let me change your view here so that you can see everybody. And please get your questions in. So let's get it started here. So a couple of quick questions for Katie. One of them came in very early. They're wondering if the duration of the study was taken into account in your analysis.
So in the part one of the presentation, we talk about variance models and how to understand variability in replicate studies. And study type is one of the input descriptors into the multilinear regression model. And so that is in some ways a surrogate for multiple features of the study.
Right.
And then we also, in the course of making 28 different models, we also break it down by study type as well. So then we'll see only the variance estimate for, say, chronic versus subchronic. So in that way, it's taken into account within that variable study type.
So there's maybe you might be answering this question also, but there's a question about developmental versus adult toxicological studies. I don't know if you compare.
Yeah, it's somewhat related. These are great questions. So developmental studies we included in the overall data set, but only the adult. So in that case, only the DAM for the systemic effects that the DAM may have experienced. We don't include the fetal generation there, and we don't include any studies where we have that sort of young offspring or juvenile. We didn't want to confuse life stages. That's another set of work I'm actually working on. It's trying to understand more of what happened in studies like multi-gen reproduction studies and developmental studies, and what are the differences across life stages in terms of what you observe and how variable it is. And that's a separate question that we didn't answer. We focused only on the adult generation of any of the studies included in our first work.
Okay. A question for George. What is meant by in vivo alternatives? and they're potentially thinking zebrafish embryos, or are you thinking other?
Exactly, some more complex organisms. Some C. elegans, for example, is another very interesting organism that can be used. We still have to learn more about how to use the data, but hopefully as we progress, we'll be able to have more case studies, we'll be able to understand better.
Yeah, okay. And I think this question is directed to George, but I think both of you might have something to say about it. They're wondering how in vitro studies can provide toxicokinetics information about nanomaterials. Because I think you mentioned nanomaterials, George, in your.
I did, yes. It's more to the understanding of the interaction with cells.
Yeah.
In terms of what the kinetics of their uptake, what is the fate? Because sometimes also nanomaterials are not always nanomaterials, so they can be they can start off as a nanomaterial, but brings them in the right environment, like a lysosome, and they will dissolve because of the pH, so they're not acid stable, for example, and then they can be handled as a normal material. But the reverse can also, we've seen examples, the reverse can also be true.
Katie, I don't know if you had something to add.
You know, nanomaterials are far outside of my expertise, but I imagine that you would be able to do analytical detection in a lot of the types of assays that inform generic toxicokinetic modeling, you know, how they partition, do they bind plasma protein? I suppose you could run the same studies. I don't know, Dr. Kass, if you're thinking of the same in vitro inputs just with nanomaterials.
Yes.
There's a couple of questions about uncertainty factor. Nicely, they're right next to each other. And so one of them is this question that I've seen talked about in different contexts, you know, is there an uncertainty factor for in vitro to in vivo essentially called for? And then this sort of related question of NAMs are using the same species if you're trying to predict effects in humans. So how does that affect the uncertainty of predictability for human health risk assessment? You know, if Katie, you want to start, and then we'll see if George.
Sure, I can start, and I think actually Dr. Kass maybe has more from his presentation. So, you know, in terms of a NAM uncertainty factor, whether it's in silico NAM or in vitro NAM, I think it's the right question to be asking, and I'm not sure if we know what the answer is yet. It's very difficult to draw a one-to-one between the set of uncertainty factors we might be familiar with versus what we're seeing in vitro. Maybe a way to extend to Dr. Kass's talk too, in terms of the toxicokinetics, you know, if you look at the high throughput toxicokinetics R package led by John Wamba, you can actually use the Monte Carlo sampler to look at inter-individual variability in toxicokinetics in terms of what is the range of doses across the human population that might result in the same serum concentration. And so you can actually now more empirically quantify that range instead of using a default uncertainty factor.
Yeah.
And so I wonder if in translating NAMs to, you know, the framework that exists, there could be some uncertainty factors that get lumped together or get separated. And then there are some cases where, well, what if you could use NAMs to refine that? So it's not a 3x or a 10x, but there's actually some empirical estimate of what the difference might be between a sensitive individual and, say, the median individual. So I don't think there's one single answer to that question, but I think it's the right question to ask.
Yeah.
Yeah, I would agree. It's a very important question. We need to get the answer right, because we have a fair amount to learn in terms of variability as well, trying to understand variability versus uncertainty. I think that is certainly one aspect to consider, and looking at the impact of knowing better the kinetics of a compound, the way it's being metabolized, but also knowing better the interaction with its little targets, and predicting then the toxicity. So I think it will take a while to learn these things. Hopefully we will not have a situation where we throw in another default uncertainty factor just because it's a NAM. That's my wish, that we use an informed approach to this. Yeah,
and I can't help but thinking, and I know both of your organizations are working different kinds of case studies and I think maybe working through different kinds of case studies will help us figure out, okay, what happens if we use this information, if we use this information in different kinds of endpoints, different kinds of approaches, trying to see what happens and what people are comfortable with, right? Let's see, there was a comment on our earlier discussion, which thank you, Shanlu Han, which mentions that nanoparticles can be radio-labeled or labeled in a non-radioactive way so that ADME can be studied. So that's a helpful contribution to the discussion. Another question for Katie, so when you were talking about histopathology predicting chronic studies, they're wondering if there's some underlying biology, or do you take into account, or do we have to take into account how long it takes for histopathology changes to occur in subchronic versus chronic studies?
Yeah, that's a great question. So one of the things that we do in this work is we group any effect regardless of the severity. And so at the subchronic level, maybe you observe a hypertrophy in the liver. And maybe in the chronic study, you observe adenocarcinoma. But we're basically saying, okay, we saw a liver effect in the subchronic and we saw a liver effect in the chronic. And so we have not distinguished the adversity, but other people have in the past. Dr. Imran Shah from our center, I want to say 10 years ago, published some work on looking at the trajectories of adversity and how in vitro Toxicology data could predict whether or not you advanced, from hyperplasia and hypertrophy to actual cancer-related findings in the liver. So, other people have made those distinctions on more of an AOP level and how do you advance through all of the severity. But in this case, we were thinking more about kind of a risk assessment approach, how you try to define a point of departure and then a reference dose that's protective for any finding. And so whether that's hypertrophy or the cancer related. So here we've made no distinction based on the adversity of the finding.
So, and there's a follow-up question actually about if you don't see hypertrophy in the subchronic, and I know you looked at negative, were there situations where you had positive in the chronic and not in the subchronic?
Yeah, so as a whole, that's exactly why we undertook that, because we thought, well, what are the odds that you miss something? And so all of the odds ratios for all of the tissues and species were less than 1. So you might be able to pick out a case example from the larger body of studies, but if you're looking at this larger database of information, the odds ratios are all below 1, suggesting that you would be unlikely to observe any finding in, say, the liver of a chronic study if the liver of the subchronic had no findings. So no weight change, no histopathology, nothing in the subchronic, kind of unlikely to then find that target organ being changed in the chronic.
We have one last question. We have a few minutes if there are any burning questions still you want to enter into the chat, folks. But so far we have this last question about whether the RefDB covers studies for volatile chemicals and if you've done any thinking or work around inhalation.
We unfortunately have very little inhalation data curated into ToxRefDB, but we're really interested in it. In the center, we have 2 human health hazard related databases that we make public, ToxRefDB and ToxValDB. And we have plans to expand at least ToxValDB, if not ToxRefDB, with more curation of inhalation study information. It relates to our interest in things like inhalation-based TTC and other things like that. So we are interested in it, but that particular type of data is not well covered in ToxRefDB currently.
Okay. One more question's come in, and maybe George will have something to say about this, given your regulatory sphere. But so it's about the use of agricultural chemicals. So use of these approaches makes sense because you have similarity in mechanisms of action and also maybe lots of data, right? How can you transition this approach to wider use of industrial chemicals?
Right, I assume the question refers to the work we're doing for the read across on the plant protection products. Yeah, please write us a message if I'm misinterpreting this.
Yeah.
So, but we'll start with that as our test case, our case study on to see whether we can, and the focus is also on repeated dose toxicity to see whether we can actually use read across for the repeated dose toxicity data, which hopefully we will be. But the work is ongoing, so we don't have too many data yet. At the moment, the contractor is putting all the data together, and the analysis should come beginning of next year.
Okay. And are you looking, so forgive me, EFSA's, you look at other types of chemicals in food as well, beyond plant protection products, is that right?
Yes, yeah, yes. So we have the food additives, the flavorings. We have, that's only, we talk each other about the food side. Then we have the contaminants, which are a major sector. It's, of course, the type of data that we get is varies considerably. One of the reasons we also went for the plant protection products is that we have the metabolites of the plant protection products, and then the main metabolites under EU law legislation, they also have to be tested. So the idea there really this would help in for the read across, to test the read across. This type of information is generally missing for food additives. They are being tested. We usually know what chemical fate or metabolic fate exists, so the ADME properties are known, but we don't have any data. Usually we don't have any data on metabolites.
Heidi, I don't know if you wanted to briefly address that question as well.
No, I think Dr. Kass has it covered.
Okay, and just to put you, to encourage you to take a look quickly at the chat, because Dr. Paul Friedman has put in a link to the databases that she's been mentioning. And I will have to close it out there. Now I see some questions coming in. I'm sorry, we're gonna have to finish up. It's at 11:30. So thank you all so much for joining. Thank you to our speakers, Dr. Kass and Dr. Paul Friedman, for taking the time to talk with us about your work. And we'll see you sometime soon at the next webinar. Have a great weekend, everybody.
Thank you very much.